Hi, my name is Dev Bhatt, number 3.3, and today I'll be talking about photosynthesis as well as oxidation of pyruvate. So we begin here with a hydrogen molecule, which is broken down via internal processes into a hydrogen. H2O is broken down into hydrogen and oxygen gas, which is seen here, as well as several electrons. These electrons go through the ETC and are powered and are charged in a sense, are excited by light through the use of chlorophyll. Here, the excited electrons power protein pumps, which pull hydrogen ions from the stroma into the inner thylakoid where they accumulate by going against their concentration gradient. Ultimately, the electron reduces in NADP plus and hydrogen into NADPH. The oxygen leaves the cell while the accumulation of hydrogen ions in the inner thylakoid go down their concentration gradient, powering the phosphorylation, essentially powering the formation of ATP from ADP and inorganic phosphate. Moving on to the light-independent reactions, carbon dioxide taken in by the cell undergoes several reactions in which it becomes fixed and usable for organic molecules. Here, ATP produced from the light-dependent reactions is used in addition to ATP plus.
Which powers ATP phosphorylation.
In addition, NADPH is oxidized, producing NADP plus, water, and phosphate groups. Through several more reactions, glucose is produced. Now, moving on.
Hey, hold on. We're supposed to not have that thing.
What thing?
That P3G5 paper. Voice product.
What do you mean?
Can you flip it around?
Yeah.
Okay. So these hydrogen ions.
So using the glucose produced from the light-independent reactions, we go here to an animal cell which can use the glucose to drive cellular processes. Here, it's broken down through several reactions, as well as using the energy from two ATP molecules to produce two NADH molecules from its respective components, and ultimately produce four ATP molecules, which is a net profit of two molecules, or two ATP. Additionally, two pyruvate molecules are produced from this process. The pyruvate molecules then go through a second reaction called the link reaction, where after carbon dioxide molecule is removed from the pyruvate, bonds to an to a coenzyme A, binds to coenzyme A, producing acetyl coenzyme A, as well as reducing NAD plus into NADH.
Okay. The acetyl coenzyme A produced during the link reaction is used to convert intermediates during the Krebs cycle. Here's the acetyl coenzyme A is converted into coenzyme A, and then during the next step, the NAD plus is reduced to NADH, and CO2 is produced. In the next step of the Krebs cycle, transforming another intermediate, the NAD plus is reduced again to NADH, and the ADP and inorganic phosphate are added together and transformed into ATP, while CO2 is produced again. In the next phase, NAD plus is reduced into NADH again, and FAD plus is also reduced to FADH2. And the cycle continues to convert oxaloacetate, to convert the intermediates. However, this does not produce ATP, but ATP is produced during oxidative phosphorylation, which
one ATP is produced, but it's in a very small amount.
Oh.
So most of it is produced in
Yeah.
oxidative phosphorylation.
My bad. Yeah. In oxidative phosphorylation, this is done using the electronic transport chain.
Oh, this one. Sorry.
Yeah. Okay, so FADH2 and NADH are electron transport carriers, which help the oxidation and reduction reactions help power the movement of hydrogen ions across the ETC. There's also hydrogen ions are also pumped from the mitochondrial matrix into the intermembrane space through ATP synthase, and as these hydrogen ions are moved down the electron transport chain. Oh yeah, sorry.
Electrons go down the
electron.
chain.
Yeah.
Hydrogen ions move into the intermembrane space.
Yeah. So my bad. Yeah. Electrons go down the ETC and hydrogen move into the space, and then these 4 hydrogen atoms are added to 2 O2 molecules to produce 2 H2O. 2 water molecules. Oh my gosh. I'm sorry. My name is Michaela. I'm 3.23.
Alright, I'll record you.
I'm Jacqueline, 3.20. Okay, so from the ETC, you have all the hydrogen that were in the intermembrane space, and they're still in the intermembrane space. But in chemiosmosis, one of the hydrogen ions will go down the gradient, well, the ATP synthase, and then you will have a hydrogen ion there, and then it will it produce?
Yeah,
It produces one ADP and a phosphate.
It produces one ATP from ADP and then the phosphate group.
Oh, sorry. Yeah, that's what I meant. Okay, so you start off with an ADP and a phosphate, and then it will transform.
Be produced.
Okay, and then an ATP will be produced from that in the mitochondrial matrix. Oh, and then after that we have fermentation. So in fermentation there's 2 types. You have alcoholic fermentation and lactic acid fermentation. Can you go to this one? Okay, so first for alcoholic fermentation, you start off with the glucose, and then that creates 2 pyruvate, which then creates 2 alcohol, and then
Via fermentation.
by fermentation. Okay, and then from glycolysis, you will produce 2 ATP. This is ADP. The ATP. You produce 2 ATP and then 2 NADH, which then goes up to fermentation, and you will produce 2 NAD plus and 2 CO2. Wait, I thought you produced 2 ATP from fermentation.
So, yeah, wait.
So then why did this end up with 2 NAD plus and 2 CO2 and not 2 ATP?
Essentially, from breaking apart I think this is drawn wrong, but it's fine. Essentially, from breaking down the glucose into pyruvate, 2 ATP molecules are produced, while the pyruvate ferments into the alcohol. Additionally, NADH is NADH and NAD plus undergo a chemical equilibrium where both of them assist in the pyruvate reaction, and CO2 is produced as a result.
Okay, cool. And then for lactic acid fermentation, once again you start with 1 glucose that transforms into
Breaks down
into 2 pyruvate, which then produces 2 lactic acid from fermentation. Okay. From here you will have, from glycolysis you produce 2
ATP.
2 ATP, okay, and 2, which goes here? 2 NADH, yes, which then once it goes to fermentation, which produces, is this where it produces 2 ATP?
No, it produces 2 ATP through glycolysis.
Would it be this one then? 2 NAD plus?
Yeah.
Okay, and then from lactic acid fermentation you produce 2 NAD plus.
Alright, great.